And now, Ask Dr. Stupid, with your host, Dr. Stupid. Hello there, I'm Dr. Stupid. Today we have a letter from Freddy, and he asks, Dear Dr. Stupid, how are proteins made? Signed, Freddy. Well, Freddy, that's quite easy, actually. See, proteins are made by the queen teen. The queen teen gives birth to all the baby teens, and the baby teens go all throughout the body. Let me put on my thinking cap. I got it! We start in the nucleus, where transcription takes place. Transcription is where DNA is copied into RNA. But how does that work, you may ask, Mr. Freddy? Well, I'm here to help. Dr. Stupid, that is. Before anything, enzymes must prepare the DNA for copying by unwinding and splitting down the middle. The two enzymes I'm talking about is topoisomerase and helicase. Think about it like some sort of beatdown. The topoisomerase straightens it out and locks the DNA in a nice position for that helicase to cut in half. Simple and PG. While that's happening, we have more enzymes help during the bullying, who tells another specific enzyme, RNA polymerase, to make some mRNA, or messenger RNA. So he goes over to the DNA and starts to copy his nitrogen bases. But he can't do it exactly. Instead of the normal adenine, thymine and guanine and cytosine, where A's and T's match and G's and C's match, thymine is replaced with uracil, but still matches with the adenine. There are some parts that just aren't necessary to the instructions, however. For RNA polymerase, it could be that the sissy parts of the DNA. The parts that we cut out are called introns, while the stuff that's kept in is called an exon. The RNA polymerase calls the DNA a sissy for not being able to leave the cell, and sends the mRNA out of the nucleus pores, into the cytoplasm, and over to a ribosome, which is like a factory to the mRNA, where translation begins. Translation is the process of RNA turning into a protein. This all takes place on a ribosome in the nucleus. Inside the ribosome, the factory workers, which are all named Trina, or tRNA, I prefer Trina, each bring in one amino acid and their own anticodons. Now, Freddy, you may ask what a codon is. I'll get into that in a second. They each bring in one amino acid, which is a building block, or also known as a monomer, for the proteins. Each Trina begins to look at the genetic code from the RNA, but it doesn't look at one at a time. It looks at 3 nucleotides at a time, which is called a codon. For each codon, it drops off the specific amino acid. For example, if one codon has uracil, adenine, and uracil in that order, it ends up being a tyrosine amino acid. For each codon, it drops off the specific amino acid after it pairs with the anticodon, which is the opposite of the codon that is delivered by Trina. While that happens, each amino acid that is dropped off by Trina are compiled into one big chain in 1, 2, 3, 4, or more. And as I said earlier, an amino acid is a monomer of a protein. We finally have ourselves a protein. Or so you would think. See, in that situation, Trina was never told when to stop until she reaches a stop codon. Trina can also do some splicing here and there too, like when it reaches the stop codon. Once the protein is complete and in that big old chain, it just has to go to the Golgi body, where it can be modified and packaged. The Golgi body is much like a FedEx quality control situation kind of thing. Finally, the protein is taken wherever it must be, oftentimes being a gene, like your eye color or hair color, resulting in gene expression, which means it's turned on, or unexpression, as I like to call it, where it is turned off. Keep in mind, gene expression is regulated. Then the cycle repeats all over again, the bullying lasts forever, and there you go, Freddy. I hope I've been some help to you today, and remember, the queen teen is mean. Well, friends, that ends my adventure. I'll tune in next time.